So the chemicals that are going to be functioning at the synapse are the neurotransmitters. Neurotransmitters, we know there's at least 50 kinds of neurotransmitters, and we're finding newer neurotransmitters by the day. In the packet, I've given you a large table of the various neurotransmitters. These are going to be released via exocytosis at the terminal boutons. They will have some sort of impact on the neuron as long as they remain within the cleft. The time they stay in the cleft is based off of enzymatic action, uptake by either the postsynaptic membrane or reuptake by the presynaptic membrane. The longer they're in the cleft, the longer they're going to have the impact on the postsynaptic membrane. There are drugs that impact the reuptake of neurotransmitters that we give in order to accentuate the actions of those neurotransmitters. There are toxins that can cause breakdown of neurotransmitters, leading to loss of functionality from those neurotransmitters. One of the things you're going to be working on is looking at drugs and substances that we might consume that can impact neurotransmitters. The neurotransmitters that we have here are the major classes of neurotransmitters. Once again, there is a larger table within the packet. Acetylcholine, ACH. This is an excitatory neurotransmitter. This is going to be the neurotransmitter that is going to be predominantly functioning within the skeletal muscle, leading to the excitation contraction coupling. It's also going to function in the autonomic nervous system, leading to a reduced functionality at tissues. The biogenic amines, sometimes referred to as the monoamines, include things like norepinephrine, dopamine, serotonin. Adenosine will also be a monoamine. These have varying types of actions based off of where we see them. Norepinephrine is going to be one of those hormones and neurotransmitters that we see involved with fight or flight. It is generally going to be excitatory. However, within the peripheral nervous system, it can have an inhibitory effect, leading to some relaxations of some muscles based off of which receptor it is acting on. Dopamine and serotonin predominantly have a central function, and they act antagonistically to each other. They can both be found peripherally. If they are found peripherally, they usually are found not technically as neurotransmitters, but as neuropeptide hormones. We have a whole host of amino acids that will function as neurotransmitters. The two that you should be aware of are GABA, gamma amino butyric acid, and glutamate. GABA is a inhibitory neurotransmitter and is involved with reduction of central nervous system functionality. It will get produced with exposures to toxins, including toxins that are ingested, such as ethanol. Glutamate is a generalized excitatory neurotransmitter. Glutamate is going to be involved with the generalized excitation of the central nervous system, involved with arousal, that is becoming awake or becoming in an alerted state. From the table on the slide where it talks about neuropeptides, one of the ones you should be aware of are the metenkephalons. Metenkephalons are going to function off of the spinothalamic tract and are going to regulate activation of the spinothalamic tract, acting as an inhibitory neurotransmitter. The metencephalons are your endogenous opioids. Their receptors are tightly regulated, and exposure to exogenous enkephalons, what we usually refer to as opioids, can drastically impact the receptors of the enkephalons. So what's the function of neurotransmitters? Well, they're going to act to propagate the signal or delay the signal. They're going to change the local rate of depolarization. They're going to change the rate of local depolarization so as to trigger the propagation or reduce the action potential trains that are going along, depending upon whether it's excitatory or inhibitory. The actions of each neurotransmitter is going to be dependent upon the chemical and its receptor of interaction. They function very similar to how hormones might function at other tissue types. Because they're going to function off receptors, modification to their responsiveness is going to be based off of sensitivity to the receptors, and this change of sensitivity to the receptor centrally leads to a lot of what's referred to as the psychodynamic effects of pharmaceuticals. Meaning based off of how the drug itself changes the receptor densities for the neurotransmitters, we can get distinct psychodynamic effects of the drugs. That psychodynamic effect of the drug can lead to dependencies and reliances psychologically to a drug that is hallmark of addictive behaviors. The way in which we're able to have some of this functionality is based off of modifications that occur within the receptors based off of neurotransmitter interactions. These receptors, even though we talk about them being a neurotransmitter receptor, are really receptors for classes of chemicals, which means that we can have what's referred to as a competitive inhibition taking place. This is where we have the wrong chemical interacting at the receptor. When the wrong chemical interacts with the receptor, it limits the ability for the right chemical to bind and cause the correct response we want. We can have what's referred to as an agonistic effect. This is where we have multiple chemicals that all have the same effect that we want coming from the neurotransmitter that we're discussing. We can also have what's referred to as an antagonistic effect. This is where we have multiple chemicals, once again, interacting on the neuron, where they are not taking the neuron towards the same function, where some might be excitatory, whereas others will be inhibitory. And what ends up happening is we have a summationing of issues coming on within the metabolic responses that we see at the neuron and at the target tissues. Additionally, we have issues that are going to come about based off of the receptors, where just like we talked about with hormones, we can have upregulation, we can have downregulation, where I either make it easier to respond, upregulation, or I make it harder to respond, downregulation. One of the things that also comes about is a change what's referred to as sensitivity. And this is based off of, does the neuron need to respond to the signal the way in which the signal is coming in? Each one of these responses, just like we saw with hormones, is going to dictate the overall functional change that we see based off of the regularity of signal and whether the neuron needs to propagate that signal down the stream of the neuronal network. So, what does this impact have to deal with in terms of how the neurotransmitter is going to function? So, what's the impact of modifications? We're going to alter the way in which the neuron is going to respond based off of how reliant it needs that secondary signal in order to have normal physiological functions, a positive or negative tolerance or adaptation, in which we're talking about how much more signal do I need with each subsequent time I see that medication. If it's positive, I need less in order to get the same response as with the first. If it's negative, I need more in order to get the same response as the first. The positive and negative impact that we see is based off of two key factors. Factor number one, what is happening to the density of receptors that I see? Are they being upregulated or downregulated? And is the drug causing competitive inhibition at the receptor that would lead automatically to an upregulation effect? A classic example of the negative tolerance is caffeine. Caffeine has a competitive inhibition effect to adenosine. Adenosine is a neuromodulating neurotransmitter. It downregulates the speed at which the action potentials are being sent. It slows down the neurons. And what caffeine does is it blocks the adenosine receptor. And the neuron, in response to that blockage, will upregulate more adenosine receptors, which is why each time you need caffeine, you have to have additional caffeine within the dose in order to get that same effect that you had on the first dose. That's not the only type of drug that we'll talk about that has this type of modification. The type of receptor being modified and the location of the receptor being modified is going to dictate the overall reliance that is necessary and the dependency that might be needed. Dependency is a physiological thing. Reliance is a physiological thing. The difference between the two is, does that physiological thing lead to a psychological thing? You can be reliant on a drug to function without having dependency on the drug. Case in point, blood pressure medicine. Blood pressure medicine, you can have a reliance on it in order to regulate blood pressure, but you do not have a psychological need for that, the dependency. The dependency is where we get the physiological responses leading to cognitive functional adaptations, where we have a psychological need and want for the drug. Most of that need and want comes from changes in receptor activation within the limbic system pathways associated with serotonin, dopamine, norepinephrine, and glutamate. Depending upon which drug we are talking about and which action we're talking about, because actions can also have the same impact, you could see a large change within the cingulate gyral area that's associated with nuclei of dopaminergic pathways. And we'll talk more about that when we discuss neurotransmitter pathways online as I discuss the neurotransmitter project.